Hello.
Hello.
Hi, how are you?
Well, I have laryngitis, so
Oh,
I won't be talking much.
okay.
I'll be back or something.
Yeah. Well, yeah, if you feel like you just type in the chat.
Okay.
And yeah. It's like it's a tempting fate, but how's your project going?
Yeah, I didn't get as many pictures as I wanted of the sound renderings because I got sick, and but I think they basically quit developing
Okay.
because they were in the fridge. So I'll get a few more pictures, but I think they're not developing anymore.
Yeah.
And I left a few in the tank, and maybe my daughter-in-law will raise a few. So, I don't know. My other project is going fairly well.
Good.
At least I got my test project going, which is that triangle. Anyway, I sound like a duck, so
Oh, it's okay.
quit talking.
At least you don't look like a duck.
Okay.
You don't quack like a duck.
No. Oh well.
Yeah. Well, thanks for the update. Yeah. So, yeah, I don't know where everyone is, but that's okay. Welcome. Well, Jyothi, how are you?
Fine.
Any updates from you?
Yeah, like I got selected for GSoC this year, so I couldn't work on that website thing, updating the UI part of DevoWorm.
Yeah.
Maybe I will complete it in June or July. I'm
Okay.
currently having exam.
Oh yeah. Yeah. Well.
No update from me.
All right. Yeah. All right. Well, thanks for the update. So yeah, Dick, did you have any updates?
Relief, we got that paper.
Oh, I know. Yeah. So yeah, we did a major revision on that paper, on the early life paper, and sent it.
Yeah. Now we see if they charge us even more.
Yeah.
Oh, well.
So I guess I don't know if people are going to come in late, but I will probably go over some community things for GSoC, and then go over some other things that I have here. So let me share my screen. All right. So the first thing I'll talk about is that we have our, of course, DevoLearn repository. Actually, it's an organization on GitHub. And this is where we do a lot of the stuff with DevoLearn and DevoGraph. So we have the DevoLearn repository and DevoGraph repositories pinned. And then we also have the models like the nucleus segmenter and some of these others that have been worked on over the application period, the nucleus segmenter, the membrane segmenter, and the lineage population models. And all of those are sort of augmenting DevoLearn, but will also be useful for DevoGraph. We also have some other repositories in here that are maybe of interest to people. So we have, actually, the DevoLearn web is a public archive. So we have, this is where we had things deployed on a web app. So we're no longer doing that, but that might be useful historically. We have some other things here as well. We have this data science demos repo, which people in the past have used to publish their own stuff that they worked on. So we've had people work on GANs models, generative models. We've had people work on networks and other types of centroid extraction and other types of models. And then we have these tutorials on data science type stuff. So people doing things in Python, and this is Shruti who pushed this. So this is something, I think I remember this. This was the neural style transfer thing that she showed in one meeting. So we have this in data science demos. So, basically in the past, we've let people contribute these and they're available for the entire community. So if you're interested, you might check these things out at github.com with DevoLearn.
Is there any link of, for example, nematode data to molecules inside individual cells?
You mean computational models or just regular?
No, not computational models. I mean molecular biology data.
Yeah, people have done a lot of like collecting data on individual cells. It's very hard to do it's not like the type of models we have where we know every cell and we have a database of every cell. We have single cell data, but we don't have, it's just kind of like the molecular data, or I think maybe there's some proteomic data. But
Okay.
but it's kind of like a database in the sense that it's a list of things. Yeah.
May I suggest at some future point it might be worth introducing, maybe through an article, the molecular biology or molecular developmental biology crowd to this data.
Yeah.
In your free time.
Oh yeah. We've kind of shied off of that, I think, in the past for a lot of reasons, but I think it should be doable. At least, there, like I said, there are always data sets coming out. They have a lot of atlases, but it's hard to really kind of use it for anything. I know that's the point of putting out an atlas, but still, it's.
Let me give you an example. The morphogenetic wave in the Drosophila imaginal disc, one we worked on before.
Yeah.
I think there may be a lot of molecular information on that. And correlating it with our proposed waves could lead to a possible breakthrough in understanding.
Yeah. Yeah, it would. It's kind of. I'm kind of hesitant to say that you could integrate the data sets because a lot of times they'll measure it in ways that are different from like the observations. So,
but they're down to imaging the trajectories of single enzyme molecules in cells.
Yeah.
Okay. So it's I don't know.
Yeah.
We need somebody who's deep into molecular biology to do it properly.
Yeah. Okay. And then, of course, we have the DevoWorm repository, which has a lot of the data sets and a lot of other types of things like our GitHub IO website and other things. So that's DevoWorm, github.com/DevoWorm, and that's a different place. But this has a lot of the stuff for DevoLearn. So this is probably the place if you're interested in participating in the community to go first. Then I wanted to go over, last time we talked about our website here, which of course is this website here on Weebly, devoworm.weebly.com. So we have a lot of things here where we have stubs that send you to different places like our Hugging Face repositories. We have some educational resources. We have a project roadmap that's kind of out of date now. We have people, which is actually quite a few people. I need to update it actually. We have different things like our DevoZoo or DevoWorm ML, a collection of Jupyter notebooks. We have media and public lectures, which is actually interesting. So I've put together a list of the media and public lectures that we've done on YouTube and mostly on YouTube, I guess. But these will go, if you go through these should kind of show the history of the project and some of the different talks that have been given, some of the different projects that we've done and so forth. So the latest one is something on digital diatoms, which is something I prepared for a general audience to kind of talk about the work we've been doing with diatoms, with image processing, and with other things. So I'm drawing from some of the publications we put out. So that should be, if you're interested in what we're doing with diatoms, that should be enough information to kind of get you started in that direction. There are other things like our work on embryo networks. Then we have updates to the OpenLearn Foundation that are kind of, they're not really timely, but they're capsules of what we've done in like certain years. So like in 2022, this kind of goes over what we did that year. Other like conference talks, posters. This is a poster of the mathematics of DevoWorm. So if you're interested in the mathematics that we use for DevoWorm, this has a number of things. So we have things, all sorts of different types of mathematical concepts that we deal with in the project, in the group, and they're broken down by category, and they kind of explain why that's important. So that's kind of an interesting approach to thinking about, like if you're coming into the group, what kinds of things are you tackling? What kinds of methods are you using? So forth. So that's a nice thing to look at. Let's see. Yeah, we just have more conference talks. Some things on open source. This is a older talk on DevoLearn, so that's like from 21. So it just kind of goes over how it started. Let's see. Yeah, then, if you're interested in making a tutorial for anything involved with the group and what we do, you're free to do that. And Ujjwal Singh, who was a GSoC scholar in 2020, did this tutorial for DevoLearn. So he went over how to run different things in the DevoLearn package. So that kind of serves as a tutorial, but really any kind of tutorial, even if it's on something that's sort of in a wide topic of the group. So that's something that people can do, and then we can put that video. You can record a video on your computer, prepare some slides, or walk through a code tutorial like this, and then we can put it up on our YouTube channel. And people sometimes are very appreciative if you go through some obscure thing. Like if you put together some code for some sort of simulation and you put it in a Jupyter notebook, and then you do maybe a quick 10-minute tutorial on it, and you go through and you show how it runs and everything. This can get like quite a few views. And sometimes you can get pretty famous for doing some really niche area of something that no one's ever done before. Like sometimes there are topics out there that no one has ever prepared a tutorial on. So it helps people walk through that sort of thing, and it brings interest to that area. So it's something that's worthwhile. It has a worthwhile component to it. So we have different talks here. We've had guest lectures too, and one we had was from George Mikhailovsky, who did a talk on structuredness and his idea of structural complexity. So he's actually done a talk here. It's on our YouTube channel. This was back in 2020. We also had, we've had GSoC students and other people give talks. And then we've also had other people like Steve McGrew, who's now deceased, who gave a talk on a microscopy method that he worked on. So this is a lot of good stuff in this area. We also have worked on other types of models like cellular automata models. And this is a tutorial on something called Morphozoic. And Morphozoic is kind of a forgotten tool. It was something that Tom Porteges developed. Tom Porteges is a senior contributor at OpenWorm. And he's done a lot of stuff. He's been working with artificial intelligence since the 80s, I believe. So he's worked on a lot of things. He's nowadays he's working on different types of like artificial life simulations, and he's got some really interesting ideas. Well, anyways, he has this package called Morphozoic, and you can actually, we have a fork to the repository on our DevoWorm GitHub account, and so you go to the DevoWorm GitHub repository, and you can fork it yourself or work with it there. And so he gave a tutorial in 2016. Last week I talked about the open house materials where we had all the projects from Devo or from OpenWorm in a repository. One of those was this Morphozoic tutorial. So in this tutorial, I talked to Tom, I interview him, and we walk through a lot of the features of Morphozoic. So he walks through a tutorial, he gives a demonstration. And what's interesting about Morphozoic is that it basically is able to recognize patterns. So you can use it as a pattern recognizer, but it also can generate patterns. And that's very important in development. So you can run a simulation where you have this grid, you have these cells that change their color based on their neighbor's state, and you end up with these patterns. So you can get these kind of dotted patterns, you can get stripe patterns and the like. And he's worked with it a little bit. He worked with it with someone, I think in the summer of 2017, and it really hasn't seen any development. So this might be something to resurrect. Since this time, of course, people have done a lot of things with neural cellular automata. And in fact, maybe I think it was in 2021, when we had Shruti in our group, and she was interested in doing stuff with neural cellular automata. This is not neural cellular automata, but this is something that's come up since Morphozoic was created. So I think it'd be interesting to have like a neural version of Morphozoic, a neural cellular automata version, to see, what it could do. Because I was really excited about Morphozoic, but it never really went anywhere. So yeah, it's another thing. It's written in Java. So if people know that, or if they can convert it to C++ or Python, and then work on integrating it with maybe some of the methods like neural CAs, that might be something for you, a project for you to do. So those are our media and public lectures. Another thing we have, of course, are publications, and so we have a lot of publications in the group. We've done a lot of work. So we've been going since 2014, and we put out a lot of papers. We have a lot of publications in peer-reviewed journals, in preprint repositories, and presentations at conferences, large international conferences, smaller conferences, and then we've of course contributed to things like some of the things that OpenWorm has done. We've contributed to a lot of book chapters. So all of these, this is the list of publications, and it looks pretty long, which it is. We have blog posts, which are posted on a blog. Those are rather informal, but they're actually quite interesting, especially with respect to some of the things we've done in the history of the project. We have infographics, which are just, visualizations of different things. We have posters that have been presented at different conferences. Some of these are tied to some of the papers.
Your video is turned off.
Oh, you mean the screen share? Let me
Yeah, I'm getting a message saying it's turned off.
Huh. Let's see. You see it now?
I see, the multiple pictures, but no, now it says video for Bradley Alicia's screen has been turned off.
Oh. Well, on my end, I don't see any problem, but.
I see it.
Okay, yeah. Sometimes it's a bandwidth thing. If your bandwidth is low for some reason, it shuts it off.
Okay, I'll shut my video off. See if that helps.
So then we have.
Oh, okay, now I see it.
Oh, okay, all right, good. So some of these posters are tied to publications and some are not. We've done this one poster neuromorphogenetic patterns and the theory of deep learning was kind of interesting because we kind of tied a lot of stuff we've done with pattern formation to some of the deep learning theory that's out there. So that was kind of interesting, but we didn't develop a paper out of it. Some of this is like a new kind of developmental biology, which is, like basically describing what we're trying to do here in the group. And then other types of, project-specific posters. One of the things about having, we have the poster online. So one of the things about having a poster at a meeting is that you see it for like maybe 2 hours and then it goes away. And it's like, we put this online, so we have it for as long as it'll be online. So people can check those out. We also have papers. We've done a lot of theory papers and simulation papers. We've done a fair amount of comparative development where we've taken C. elegans and compared them with different species. So we have one paper where we compared C. elegans and zebrafish, and another where we compared C. elegans and Ciona intestinalis, which is the sea squirt, which is a marine invertebrate with a very interesting life history. So that's, where they go through different larval stages. So those are interesting comparative biology papers. We've, of course, done work on the diatoms under this Basilaria category. Drosophila, which is the fruit fly. We've done a couple of papers on that. And then, of course, C. elegans, which we've done papers, and we've generated data sets on that. So those are all what we have for our publications. And so I want you to, if you're involved in the community period, or even if you're not, I encourage you to take a look at those things. It'll give you a better handle on where we are as a community and what we've done in the past. So it looks like Sushmanth is here.
Oh,
Hello, Sushmanth.
Hi there.
Do you have any updates for us?
Yeah. I have my updates. I was completely working on this week. I was completely understanding cell tracking channel data set because there are different files in it. So I plotted them due to some image, different image shapes, etc. Some images are not plotting, and some things have happened. I'll show it to you.
Oh yeah. All right.
This is the file of whole thing. I plotted every file in it of cell tracking challenge data set. I was saying like what are there. I wrote a clear commented code. These are outputs of it. And sorry, last meeting I told this is like some kind of bars of the images. No, these are the compression laws of the image. When image is converted from TIFF file to JPEG, because when you convert a TIFF file to JPEG, there will be a compression in the file also. So these are the data points which are more compressed. So these are here which are that is less compressed.
Oh, okay.
And I tried to read the documentation of PyPlot. Then I was able to understand what this graph represents. So I use it. I just converted a TIFF file to GIF and I tried to plot it how it looks. Then I'm just seeing like whether can I use this data to train a 3D nucleus segmentation for video tutorial. So the video is looking fine. These dots are the centroid position of ground truth position of each cell. So I could use this and I converted them. I'm seeing like some of the files are not plotting actually. That's due to some data missing, etc. And I started almost coding about this one, what we call fine-tuning SAM. I found different resources to actually fine-tune using segment anything model to integrate with DevoLearn. This is from a tutorial from a Hough page. I tried to use this, but there are some of the other reasons I stopped using it. And I got another course that is mainly microscopy methods, fine-tune a medical data set using some, and you can expect the weights from the using this the way to use last couple of last configure which library should I use. I'm using countries because I have two options here, and there is another option also. Actually, Mayuk and Mainak used this library to segment their models in PyTorch, and there is a pull request here for SAM, integrating segment anything model as an encoder for this library. It was never merged into their code base, but there is a pull request. I thought this would work, and I wasted around 2 to 3 days using this library, but at the end I came to know that this code has little bugs, so I couldn't use it. Then after that I could find these two resources to use. So I was thinking to start working on the code as usual. If I complete this, I will first use this segmentation. Here I could use big data set for that, and for the nucleus segmentation, this is the how game gonna go. Like first I will segment each nucleus. After that I will find out the centroid of each cell, and using OpenCV library I will try to calculate the volume of each cell. That's how I think I wasted a lot of time on this library, like making it like even talk with some of the functions like how should I use it, etc. It never worked. I wasted around 3 days. I don't know where I went wrong.
Huh. Yeah.
And these two libraries right now I'm using this MedSam to segment, and I started working on the DevoNet, the proposal which I proposed in my proposal also. I have found the dataset for to segment these nucleus. The cell tracking dataset is perfectly used because as you see here, we have a nucleus segmentation and ground truth for each nucleus, and as well as we have the centroid detection method. So I'm going to use this to try to use each centroid and volume, mean area, etc. can be calculated. I'm just updating the whole code. Actually, I have written the code for it, but it is in the older versions of Python, like Python 3.8 and 3.7. I'm just upgrading it to Python 3.10.9 and scikit image newer versions. And that's not my update is. Right now, I'm just confused like which library to use. Last night, I have talked to you. Then he told like use this MedSam library. You can fine tune the whole thing. And here we have to implement this also. Here we have some video tutorial etc. to see and implement it. So that would be not a big problem. That's what my update is.
Okay.
Okay. Sushmanth, I got a general question. For all of these where you have done the segmentation of sections, I gather, can you display them as three-dimensional objects and perhaps put them in rotation so that they are perceived three-dimensionally?
Actually, for annotations part, we don't have the ground truth actually, like mentioning the annotation, this is a cell. I will just take that as a cell. I'm giving that as only one class was there because only one cell, only cells are there in that image. So I'm going to use that only as an annotation. We can mention that in the code, and most of the part can be handled by segment anything model because it is over 11 billion images. So even the parameter of our network was 696 million, which is the largest.
Okay, I'm not sure we're not understanding each other. Can you go back to an image of your section data?
Yeah.
Show me
This one?
You gotta share your screen.
This one, or I'm sharing my screen, right?
No.
No.
Oh, sorry.
All right, there we go.
Yeah.
Okay. Yes. Okay. Those are fine. No, these are sections, correct?
Yeah. I mean, in a TIFF file, these are like different video frames. This
Right.
is a
But from the point of view of the embryo, there's sections through the embryo, right?
yeah.
I think
Okay,
they're all the
can you-
same section, though, aren't they? Or,
Can you prepare a display which shows them perpendicular to this view?
Perpendicular to this view. Okay.
So a single section would be a plane.
Okay.
yeah.
And then those cells which appear in multiple planes near each other give you outlines for the 3D structure.
Okay.
Okay? And then you could display it in 3 dimensions by taking the whole structure and slowly rotating. Slow rotation gives the optical illusion of 3 dimensions.
Okay. I understood. A 3D structure to view this whole 3D data set. That's you really want. I mean, that's the thing you want, right?
Yes, right now it would look like a bunch of little circles, but if you connect the circles, you get volumes.
Yeah. I understood. Okay, now
Okay.
I got that. I will work on this. That would be a great addition for this to understand it clearly. I think we can use image J for that to visualize it. So I will try to use image J and plot it in a 3D structure, and I will show it by next week.
The other thing which can work is to make stereo pairs. And there are now stereo viewers that you can put directly on your computer screen.
Okay.
So you make a stereo pair, you observe one through your left eye, one through your right eye, and it becomes 3-dimensional.
Okay.
I don't know. You can buy these things. I've got one on order from Amazon. Stereo viewers, and they're made for monitors. You can get an inexpensive version of this for, well, it was $9 Canadian. I don't know what that translates to.
Yeah.
Okay.
Okay. I'll definitely see. And I will try to plot it in 3D structure as you mentioned. Actually, I worked with this before, like on different cells, mouse cells actually, to plot them in a 3D structure.
Oh, okay. Okay, good. Then you know what.
This would be not like big problem. I will try to create a 3D box around these cells, so it looks like a 3-dimensional in different view. If we change the view or if we move the mouse, the cells will move a little bit, and we can see that 3D effect, like how the cells are moving.
Right, you rotate around it. Now you can also do that with the centroids.
Okay.
That might be easier because the centroids are more accurately defined.
Okay. And this is the, I think this one is the centroid thing. Where is it? Active to GIF conversion. Yeah, this one. This is just a 2D view. I'll try to create a 3D view for this also.
Yeah, exactly.
Okay.
That would work very well with stereo pairs.
Okay.
Or with rotation, either way.
Yeah.
Okay. I think there are some specific libraries for this kind of stuff in Python. We can use them and plot them. So I will do it. This is not a
Oh, okay. If you find them, please send me the URLs for them.
Libraries also. Okay. I think the best library we can use is ImageJ or Fiji. I know this library because of you only. You have mentioned it to Mainak, the last year GSoC student. He mentioned it to me.
Right.
Like you can use ImageJ to.
There's a there's a new version of ImageJ. It's called Fiji.
Yeah,
F-I-J-I. Okay, which I haven't used yet, so I don't know anything about it. It doesn't exist.
Okay, I will try. I can do it. Just 3D plugin is there. We need to keep the 3D
Okay.
plugin.
The reason I'm asking is for a very strange thing. I'm working on a paper with two ladies in India on observing galaxies in 3 dimensions.
Oh.
Okay.
Okay.
It looks like it might be possible.
I will try to implement. Bradley, those are my updates. By next week, the weight extraction for each model can be done, or converting it to ONN part, like the model. I mean, the Hugging Face spaces for instant segmentation can be done. I'll start working on it. Maybe it will take 2 weeks or so, but I'll try to complete this week only.
Okay.
Yeah.
That sounds good. Thank you for the update. Yeah, looks like interesting stuff, and looks like, I know you're stuck on some things, but that's part of what we're doing in the program. Trying to figure out how to get around some of these bottlenecks, and if you have like some other things you can do in the meantime, then that gets done, and you just kind of work on that problem. So I think that's a good demonstration of that.
And another thing, can you extend my GSoC period till the last 22 weeks, because I want to take like as much time I can to complete this project. Is it possible?
Well, I think you can get an extension, but I think you have to wait until sort of towards the end to see if you can get it extended. The only thing it does is like it just basically you spend less time on it per week. You know what I mean? Like it's the project length doesn't change. It's just the amount of time you have to complete it. So if you extend it, you get fewer hours, or you don't get paid anymore. Yeah.
Last week, Stephanie Taylor, we had a meet with her actually. She invited all GSoC contributors to it. She mentioned like if you want to increase your timeline, mention it to your mentor beforehand, because you need to make changes in some web app to know them, like my project has been extended. According to that, only funds will also be released. My first evaluation timing is going to be changed from July 14 to some other period. She mentioned something like that. That is the reason I'm mentioning it. I just want to work on this clearly, perfectly. That is the reason I was extending time period.
Okay.
Make perfectly clear.
Well, if you could write up a rationale for it, like why you want it extended, and I can contact, because I have to contact the people at INCF, and I think they are the ones who do the grant, or I guess GSoC grants the extension, but through them. So it's like I think it still probably would be like later on in the summer where we say that. So we'll see.
Okay.
Yeah. All right. Well, yeah, thank you for that update. That was very good stuff. And it looks like you're getting into a lot of good toolboxes and things like that, and we're moving the ball on that. So, and we're still technically in the community period, so that's good. We have a couple more weeks before the official coding period starts. It's good that people are getting their ducks in a row and everything. So that's good. So now I'm going to talk about some things that I found. So I think one of them is actually from the Slack channel that we had, and another thing is something that I think will be interesting. We were just talking, for example, about genetic data and gene regulatory data, and last week we talked about engineering mammalian cells, basically. So there's this field, synthetic biology, where people they can boot up cells to do certain things. So people have done this extensively in bacterial cells. We have to boot the cell up, and they can do something with the bacterial cell. It could be like some industrial use or some other type of use, maybe for medicine. Mammalian cells are a lot harder for a number of reasons, but the thing I showed last week talks about sort of the toolkit that we would need to have to build, to engineer mammalian cells. But one of the other things we need is to have methods to manipulate genes, because genes, of course, are the building blocks of proteins. Genes get transcribed, and oftentimes what happens in a lot of the experiments you'll see in biological papers is where they manipulate a gene to do something else. So there are methods, and these go back many, many decades, where they would mutate a gene, or they would manipulate it in some way. They might knock it out. They might knock out the gene. They might what they call overexpress the gene, which means it gets expressed over what it would be in normally. You can have multiple gene copies, so you can insert them into the genome in different places. You can do all sorts of things like that. But in mammalian cells and like in animal cells more generally, there's been a lack of control. In other words, you can put the genes in or knock them out, but sometimes you don't really have good control across cells. So you have to do a lot of selection of cells that are expressing the gene in the right place and so forth. And so gene editing is actually an area that has been sort of elusive but moving fast. There have been a lot of things like constructs where people would go in and they'd create these constructs where they design a way to put genes into the genome. And there's been a lot. You spend a lot of time designing like the right vehicle to get genes sort of into the cell and then get them into the genome to do things like overexpress them or to replace the function of something else. And so that's been a thing. And then recently, maybe in the last 10 years, we've had what we call CRISPR, which is a technique where you use zinc fingers to open up different parts of the genome, different parts of the chromosome, and pull out genes or insert genes. And so this is something that has been, people have been perfecting over that time. I wouldn't say it's perfect, but it's a lot more accurate than some of the earlier methods. So this technique that I'm going to talk about today is sort of a step up from CRISPR. And I'll say right now that CRISPR is like this it's hard to wrap your head around some of the terminology they use. But basically, they're selecting, they're going down the chromosome, at the nucleotide level. They're finding certain stretches of DNA. They're targeting that, and then they're either excising it or putting something in. So it's very precise with respect to the position of a certain sequence. And so this has led to a lot of medical advances, at least in theory, because they haven't really used CRISPR in humans. They've used it in plants and some organisms like C. elegans and other model organisms. This is something called prime editing. And so this is like a step up from CRISPR. And so prime editing is actually, like I said, it's gene editing. You can go through and you can edit out genes. You can edit out stretches of DNA. You can insert new stretches of DNA. And so in that sense, it's very highly controlled. You can do this across cells, so it's replicable. And then you can use it to maybe treat diseases. And so they talk about disease here, but a lot of times in these articles, when they talk about treating diseases, it's kind of theoretical because they can't necessarily do it like at the clinical level, but they can just show the proof of concept. So this is prime editing shows proof of concept for treating sickle cell disease. So sickle cell disease it affects red blood cells, and it's a serious blood disorder. And it can, they've been trying to find cures for things like sickle cell, where it's largely driven by genetics for many, many years. And it's one of these things where, a lot of people are really are looking hard for cures, and they don't come easy because one of the things you have to do, of course, is to edit the genome to, at least overcome the genetic aspect of it. But also to have to deal with how the genes are expressed. So, this is where precise genome editing is very useful. So this group from MIT, the Broad Institute, St. Jude's Children's Research Hospital, they've showed that a precise genome editing approach, prime editing, can change mutated hemoglobin genes back to their normal form in SCD patients, which restores normal blood parameters after transplantation into mice. So they do a lot of things with the cells, to move them from, sort of the clinic where you're modifying the genes to the human. And so they have to do some things with the cells, to get it into the form where you can actually use it for therapy. So it's kind of an interesting field because they're always trying to figure out ways to do therapies. And again, this isn't something that's approved by the FDA. It's kind of like these proof of concept things. So this was published in Nature Biomedical Engineering, and it's just an example of this prime editing technique. So the prime editing technique is, let's see if they talk about it here. Actually, maybe not in this paper, but this is just the paper where they show the application to sickle cell. And then so the prime editing approach is a promising approach because in theory, we can directly correct disease mutations to specify healthy DNA sequences we're choosing. We optimize prime editing in long-term blood stem cells and show that the prime editing cells maintain full engraftment efficiency in an animal with a clinically relevant system. So they're able to fix mutations using prime editing. They're able to take this system and they can find the disease-causing mutation. We have our, the human genome sequenced. We can sequence genomes more and more efficiently, so we know kind of the positional locations of these mutations. And so we can use these techniques in tandem with that information to fix specific mutations. So this is all coming together to sort of treat diseases. So you can replace these mutations with high efficiency. Prime editing successfully corrects this mutation with up to 41% conversion in blood stem cells from patients. Previous research has shown that editing in over 20% of cells likely translates to therapeutic benefit. So, if you go through all the red blood cells and you just make these edits to the cells, and this is would be a high throughput procedure, you could actually treat and cure the disease. But you have to have a system in place where you could cycle through the blood. It's almost like filtering the blood. Instead of filtering it, though, you're fixing the genetic mutations that you want to improve upon. So improving precision gene editing. So this is just kind of talking about how it's sort of the origins of this method. So the way they do it now is they use Cas9 nucleases, which make double-stranded breaks in DNA that prime edits largely avoid. And the reason they avoid that is because there's some problems with that approach that have to do with the stability of the DNA, I believe. But the collaborators have previously shown base editing, an alternative genome editing technology, can turn mutations into a benign variant. But that doesn't happen in the original healthy sequence. So this is in a Nature publication where they talk about this, where they're able to treat this in mice, and they're actually looking at the function of the blood cells with respect to the mutation. And so, yeah, this is just heads up on a new type of technique that is being developed. So this is prime editing. It's sort of an advance on CRISPR-Cas9, which we may run into in the literature. And so that's one of these cutting edge techniques. So, hi Morgan, how are you? I think speaking of cutting edge techniques, that's what Morgan likes to hear about. Great. So that's all I have to talk about for that. And then this other paper that came up in the Slack, it's I think in the DevoLearn Devo Graph channel, or I'm not really sure where it is. It's in one of the channels, either that or the DevoWorm channel. This talks about harnessing the modularity of cells to program complex biological functions. So this is going back to the cell engineering idea, where we want to like engineer cells. We want to improve their function. We want to change their function. And to do that, though, in mammalian cells, say, we need to know a lot more about how cells work. And we don't fully know how cells work, but we can use some of the principles that we've learned from, say, systems biology to do this. And so one of the things we've learned is that cells have this aspect of modularity about them. So, this is something that we can use to program these complex functions. Last week, I talked about the different ways we can do that. But this week, I want to talk about this idea of modularity. So a new era of biological engineering is emerging, in which living cells are used as building blocks to address therapeutic challenges. These efforts are distinct from traditional molecular engineering. Their focus is not at optimizing individual genes and proteins. So this is what we talked about in the case of prime editing, but rather on using molecular components as modules to reprogram how cells make decisions and communicate to achieve higher order physiological functions. So this is the cell-centric approach as opposed to the gene-centric approach. And so they both have their place, I think. Modifying genes is good for treating diseases and targeting specific functions where you know what the underlying genetics are. If you don't know that, then the cell-centric approach makes more sense. And so the cell-centric approach has a growing toolkit of components that can synthetically control core cell-level functional outputs, such as where in the body a cell should go. This is the stuff that Michael Levin does in some of his work with flatworms, trying to think about like the whole organism and sort of how cells can, regenerate and find a place in the organism. And so, but people have been thinking about this for a long time. This is a very common problem in developmental biology, for example. But we can do this, either by sort of letting nature do its thing and then assisting nature, or we can actually design systems that would allow that's sort of where we can control this. So maybe we can put cells in places where they wouldn't normally go. So if you're doing stem cell therapy, for example, you have a stem cell that you're differentiating, and it normally may not know where to go or go in the right place because it may derive from, say, some other type of tissue. If you know the rules of the system, you know how to guide those cells into the right place. So this is the type of thing we're wanting to do here. So talk about how this can be used by forcing the conceptual distillation of complex biological functions into a finite set of instructions that operate at the cell level. These efforts also shed light on the fundamental hierarchical logic that links molecular components to higher order physiological function. So they talk about modularity here. They talk about this sort of set of instructions. So this is a graphic here where you have a change in the genome and molecules. And so this is the DNA here. These are the membrane proteins and other types of proteins that get formed that do things in the cell. Then you have things at the cellular level where you get inputs from other cells. There's this decision-making circuit. When they say decision-making, they don't mean like the cell thinks. They mean that it either does one thing or another. And there are all sorts of different genetic circuits that allow it to make these. So in other words, the cell can decide to live or die. And so the cell can either go apoptose or not. That's a decision-making thing. And so there's a change there, and it's triggered by some input from other cells. It's something that's done through genetic regulatory circuits. And then that's the output to other cells. So there's this effect from cell to cell. And then you have these multicellular networks which interact so that you have, things like in the retina perhaps, or in the brain, where you have, different networks that interact and they change their physiological state in that way. So then you have this cell state toolkit that then allows you to manipulate these cells so that they can do these things. You have a toolkit that allows you to transmit signals, send signals, move and change its shape so the cell can change its shape. We've talked about that a lot. But to have control over that, is the thing that they're proposing here. Death, where the cell dies, differentiation, proliferation, and adhering to a surface. So the idea is you have these modules, they think of this as modules. So, a transmission module would be something we can engineer and say, okay, we want the cell to transmit a signal at this time, or we want the cell to not transmit signals. We want the cell to change its shape in a certain way. So we have this toolkit. And this is, again, I think this is more kind of a aspirational thing. So they don't propose an actual set of tools, but they say that you have this toolkit, and ideally it's like a artist palette where you have different colors, color gradations, and you pick one and you put it on, you turn it on and you see if it works. You try another one to see if it works. And then you move to another function and you turn it on. So this very much assumes that these functions are independent in some ways, that they don't interact, which is maybe wrong. But that's the idea of modularity here. So yeah, we want to have this universal toolkit, and the whole idea is basically being able to push buttons and get outputs that we can, basically control like a button press, which maybe, again, like in biology, maybe that's wrong-headed. Maybe that's fundamentally misguided, but that's the goal here. So, we have these, we actually know that there are, genetic regulatory networks for certain functions. We know, like in some diseases where we have specific genetic mutations that turn diseases on or off or modify them in some way. So we know those exist, but we don't really have a toolkit for that. And sometimes for some diseases, it's a little bit easier than, say, other things like a change in shape. Could we say that, if you just change these mutations or if you knock this gene out, that it changes shape? I mean, there are a lot of papers where they do these kind of experiments, but the question is how accurate do you get? I mean, can you really control that? And so that's the idea. And then from that toolkit, you can then go and have a customized cell response. So we might be able to, say, recognize diseased tissues. And when you see a cell that's diseased versus not diseased, you can destroy that cell, turn it off, and say die, versus the healthy cells, you can say you need to live. And you can push that button. And the idea would be that you'd be able to replace, say, cancer cells with normal cells. And so, or favor normal cells over cancer cells. And you also have engineered multicellular networks. So you have these kind of networked interactions between cells that you could also control. And so this kind of goes through some of these things where you have these cellular tasks, like delivering suppressive payloads, identifying inflammatory sites, identifying and killing cancer cells. So there are all sorts of things that you might want to do. And then they have this, what they call the cell chassis, which are, at this point, I guess, just kind of lists of cells that you could use. So NK cells, T cells. We have a lot of different cell types that we've developed in the lab. And so those, are useful for kind of driving this forward. So you wouldn't use like normal cells. You'd have to use something that was engineered for the purpose. And so, some of those are, candidates that we can engineer. Some of these, like NK cells, are very specialized cells. So, these are, this is moving towards a toolkit, I think. And they show that there are all sorts of different potential therapeutic areas of application, from synthetic development of organs to regenerating systems, to reducing neuroinflammation, in reducing neurodegeneration, reducing inflammation, and destroying cancer. So these are all, novel goals, but I don't know if we're really kind of anywhere near this. So just to keep that in mind. So this quote says, engineering higher order functions forces us to grapple with the underlying logic and hierarchy of biology. And indeed, there's this, like, we know a lot about sort of that, we know that biology is hierarchical. We understand there's some logic, especially in terms of genetic circuits, but we don't really know the intermediate area, how that all fits together. So is that something we learn through engineering or through like theory and experiment? It's hard to say. So that's what this paper is about. Thank you to Jia Hong for pointing that paper out. It was very interesting. Yeah. So any questions or comments?
You've got a link. Yeah, sorry.
Oh yeah, I'll put it in the Slack. All right, now I'd like to talk about two additional papers, and these have to do with cell cycle and the role of something called transcriptional filtering. And so this is involved in cells building tissues. So the first paper is from eLife. It was published in 2021. And the title of this paper is Control of Tissue Development and Cell Diversity by Cell Cycle Dependent Transcriptional Filtering. So the authors introduced this term, transcriptional filtering. So the abstract reads, cell cycle duration changes dramatically during development, starting out fast to generate cells quickly, and then slowing down over time as the organism matures. Our group has explored that in the context of C. elegans and zebrafish. So this is a general phenomenon that happens across species. The cell cycle can also act as a transcriptional filter to control the expression of long gene transcripts, which are partially transcribed in short cycles. So in this paper, they use mathematical simulations of cell proliferation, and they identify an emergent property that this filter can act as a tuning knob to control gene transcript expression, cell diversity, and the number and proportion of different cell types in a tissue. So this is something that, in cell division, where there's a control mechanism that driven by cell cycle and the length of cell cycle that produces different size transcripts. In the short cycles, these long gene transcripts are partially transcribed. Cell division starts to slow down. Then you get these long gene transcripts transcribed in full. So basically, you're not getting all the information you need early on, so that might impact function. So this actually acts as a filter. So we generally think of cell proliferation as this thing that controls the number of cells in a tissue, or maybe the size of a tissue. But there's also the secondary aspect where it's actually producing transcripts differentially. So they use mathematical simulations to sort of identify some of these things. It acts as a tuning knob, so if you change the length of the cell cycle, you can change this aspect. It's maybe an epiphenomenon or a secondary effect, but it does have an effect on the overall tissue. Our predictions are supported by comparison to single-cell RNA-seq data captured over embryonic development. So they're using RNA-seq data to sort of confirm this. So this is where you have sequencing the transcripts and looking at their sequences and seeing how much is present or absent in a certain cell. Additionally, evolutionary genomic analysis shows that fast-developing organisms, which means organisms that have a shorter development, where they have a quicker pace to certain tissues, if the time it takes to build, say, like a wing or some other structure shrinks, which happens in development, then this can affect this as well. Evolutionary genomic analysis shows that fast-developing organisms have a narrow genomic distribution of gene lengths. So if it's a fast-developing organism and it has these short cell cycles, the gene lengths are very sort of concentrated into a certain set of lengths, and so that means a certain set of transcripts are being functionally expressed. While slower developers, meaning that they go from this short cell cycle to a longer cell cycle because they have a longer pace of development, has an expanded number of long genes. So it means that longer genes are expressed in these organisms, that longer transcripts are longer genes are fully expressed into complete transcripts, and that can have an effect on function. So there's this concept called heterochrony, which is actually a very interesting concept for morphology, because the idea is that you have a growth trajectory, and sometimes that growth trajectory can be increased, so the rate of growth can be increased. Sometimes it can be decreased. So you start in early development and you decelerate growth meaning that, you shorten the time to maturity, or you shorten the growth rate, and you end up with, say, a smaller organ or a smaller organism. And so that's how we generally view changes in development across evolution, that there are parameters controlling that growth rate, and that those parameters change. And as those parameters change, the morphology changes. It operates on the same basic architecture, genomic architecture, but it just changes how the genes are expressed, how the organ is, how long the organ has to grow, and so forth. So you see this with like brain growth in mammals. You see that different parts of the brain grow at different rates. If there's a longer developmental period, the brain grows to a larger size. If growth is accelerated, it grows to a larger size, sometimes structures within the brain, sometimes the brain itself. And so you'll see this across the tree of life in different ways. And what this suggests is that these heterochromic parameters have sort of an effect on the tissue and ultimately affecting cell cycle, but that there are transcriptional consequences to this. And maybe those are important in selecting those parameters across species. Our results support the idea that cell cycle dynamics may be important across multicellular animals for controlling gene transcript expression and cell fate. So this is, definitely they're proposing this mechanism, and I think it's an interesting proposal because I think it's probably going to have some sort of an effect. What the actual effect is on growth and development, it's not quite clear, but we can see evidence of it in the data. So a fundamental question in biology is how a single cell eukaryotic cell, or a single eukaryotic cell, zygote stem cell, produces the complexity required to develop into an organism. A single cell will divide and generate many progeny, which means they generate a lot of different offspring cells or daughter cells, diversifying in a controlled and timely manner. So we know that cell cycle is, the length of cell cycle involves basically producing or proliferating cells. And so this is something that we see with in all sorts of ways in development, from generating cells for stem cell crypts to generating cells that differentiate into tissues. And so when you need a larger tissue, you generate a bunch of progenitor cells and they differentiate into the cells that will form the tissue. The more cells you have, the larger the organ. So these cells are generated with very different functions than the parent, all with the same genome. Many regulatory mechanisms coordinate this process, but much remains to be discovered about how it works. Here we explore how cell cycle regulation can control gene transcript expression, timing, and cell fate during tissue development. So cell cycle has 4 phases. It has the first gap phase, G1, the synthesis phase, S, the second gap phase, G2, and the mitotic phase, M. And of course, in mitotic phases, when it actually divides, and you go back to the gap phase for another round of division. So you can see that there's these four phases of cell division, cell cycle. So it goes from dividing to replicating its DNA and then dividing again. And so the idea here is that there's a control on these phases, and there's a whole genetic circuit that's involved in this where it regulates the timing of it. So if you shorten the first gap phase, you can shorten cell cycle. If you shorten the synthesis phase, you can also shorten cell cycle. If you lengthen the synthesis phase, you lengthen cell cycle. One of the consequences of that, though, is that you lengthen the time in which these things can take place. So you might have things going on during that time that are, have consequences. So for, an elongated synthetic phase, that not only has an effect on the entirety of cell cycle, but also has an effect on what's going on during that phase in the cell's metabolism. So the length of each phase determines how much time a cell allocates for the process associated with growth and division. Again, this is a timer basically divided into 4 components, And if you lengthen any of those components, you can lengthen cell cycle. And lengthening cell cycle, as we've said, has a number of effects. However, the amount of time that is spent in each phase frequently differs from one cell type to another within the same organism. So different cell types, mature cell types, stem cells, they have different amounts of time. So their cell cycle is different. This is true, I think, for stem cells, but also for differentiated cells. And you can definitely have, a change in the cell cycle for depending on the function of the cell. So this is kind of what led them to this study, to think about like, okay, why is it that you have this change across cell types? We know there's this general trend in development, but we also have this variation among cell types. Organisms such as the fruit fly and the worm, Drosophila and C. elegans, respectively, exhibit cell cycle durations as short as 8 to 10 minutes. Cell cycle duration also changes over development. For example, it increases in mouse brain development from an average of 8 hours at embryonic day 11 to an average of 18 hours by embryonic day 17. So there's a shortening of time. There's this difference in time across cell types, but there's also this difference in time across organisms. And some of the interorganismal differences have to do with the life cycle or the length of their lives. So fruit flies and worms, for example, will only live a couple of days, whereas mice will live a lot longer. So their cell cycle can be longer. But still, you have this great amount of variation in cell cycle across cells, animal cells. So that's interesting, and really it does have a number of consequences, and probably we're not talking about some of them. So interestingly, cell cycle duration can act as a transcriptional filter that constrains transcription. In particular, if the cell cycle progresses relatively fast, transcription of long genes will be interrupted. In typical cells, the gene transcription rate is between 1.4 and 3.6 kilobases per minute. So that means it goes down the DNA strand and it transcribes something like 1,400 to 3,600 bases per minute. Or I guess it would be, yeah. So it would be, well, I guess that, that would be right. So this is a pretty fast moving polymerase. So you're getting a lot of things transcribed per minute. But if you think about it, that rate doesn't necessarily change. You're just transcribing more or less. So if your cell cycle is shorter, you get less transcript. Now, that's a pretty large range. So that's, we don't really know how that varies. Maybe it varies with cell cycle. So you get some compensation for a shorter cell cycle. So in other words, it goes faster with a shorter cell cycle and maybe slower with a longer cell cycle. But I haven't looked at that reference, so maybe that's not the case. Thus, an 8-minute cell cycle would only allow transcription on the shortest genes on the order of 10 kilobases measured by genomic length, including introns and exons, whereas a 10-hour cell cycle would allow transcription of genes as long as a megabase on the genome. So this just shows you cell cycle duration changes during mouse development. So this is the different cell types in mouse, from embryo to trophoblast to mesoderm to retina, neural tube, cerebral wall, cortical stem cell. Just a large, actually retina's over here. So this is a large variety of different cell types, and this is the number of days in which you have. So this is the cell cycle duration here in hours on the y-axis. On the x-axis, you have days of development. So you see that the embryo is pretty early in development, embryo cells. You have the different germ layers here. You have retina cells early and retina cells late, retina cells in the middle. And you can see that the retina cell, if we look at the retina cell and we track it across time, at 7 days of development, the cell cycle duration is very short, and it increases from about 4 hours to about 11 hours from day 7 to day 10. And then to day 15, it's about 20 hours in duration. And then actually at day 22, it's about 30 hours in duration. So you can see that there are actually 4 data points here. That's a pretty good trend of like over developmental time and increasing. Cortical stem cells, on the other hand, it's less clear. You have a bunch here at about the same time in development, and they're kind of varying in the number of hours for their cell cycle duration. That's actually probably not surprising since the stem cells are pluripotent, and so they can form a number of different cell types, whereas retina is already differentiated. In any case, this is what it looks like. You get this tendency towards, as you get further along in development, of higher cell cycle durations, or larger in the number of hours. These embryo cells are actually an interesting exception to the rule. For some reason, they're around 20 hours of cell cycle duration, but it happens very early in development. So I don't really know what's going on there. Again, it may be because they're pluripotent or even totipotent. We don't really know. But in any case, so we asked what effect cell cycle dependent transcriptional filtering may have over early multicellular organism development. Through extensive mathematical simulations of developmental cell lineages, we identify the novel and unexpected finding that a cell cycle dependent transcriptional filter can directly influence the generation of cell diversity and can provide fine-grained control of cell numbers and cell type ratios in a developing tissue. So what that means is that you have this change in cell cycle timing, and that cell cycle timing can control sort of not only the number of cells that are proliferated, but what they express. And so if you can control what they express, or if there's filtering of that expression in terms of short transcripts versus long transcripts, then you can affect the types of cells that are in your tissue. And this is not a conscious thing, but this is something that happens just through this switch. So there's a switch that happens, or some sort of gradation that allows for this kind of control. And so, again, we have this. So this is cell cycle duration. This is a cell with a set of transcripts being produced. The gene length is the introns and exons. So it's like the coding regions and the non-coding regions all together. And this is actually an interesting point, because the longer the transcripts, the more things that are being produced. So in other words, you'll have a lot of opportunities for alternative splicings, for example, where the RNA is spliced in different ways to take different parts of different introns from that gene and use it for different things. So it has a sort of enhanced regulatory capacity. When you transcribe shorter gene lengths, you're only getting part of the gene. Perhaps you're only getting the sort of the main function of the gene. It's not really clear to me, and I'm not an expert at like the structural biology of genes, but what that consequence is, or if it's even something that's predictable. But the idea that you're putting out introns and exons means that you have this opportunity for alternative slicings or alternative isoforms of different genes. And so this allows you to have an enhanced function. So this shows that the RNA-seq, so you're actually looking at some of these, you have different isoforms and you have different genes, and you can actually get the transcript counts. This is basically what they're doing in mRNA-seq. So they're actually looking at the transcript count for gene 1, and gene 2, and gene 1, gene 2, and gene 3. So basically what happens here is that when you shorten the time that your cell cycle happens, you're only getting maybe one gene that's being transcribed from this circuit. Because 1 is shorter than 2 or 3, 1 is there, but 2 and 3 are not. If you lengthen the duration of cell cycle a bit more, you get the second gene that pops up, but you also get 2 isoforms of gene 1. And then you can see that here. And then you not only get, if you lengthen the cell cycle even further, you not only get all 3 of these genes in the circuit, but you also get 3 isoforms of 1. And it could also be 3 copies of the same transcript. It's, this is the thing about like lengthening the window of transcription is that in this way is that you get a lot of diversity. You not only get more genes, but you get more types of genes. So you get maybe more transcripts of the same thing, or you get more isoforms or whatever. And so you can see that this happens to affect cell division, because when you have a longer cell cycle, you get more transcripts that can be randomly distributed to the daughter cells. And that leads to different types of transcripts being transmitted from mother cell to daughter cell. And it leads to differentiation in different ways. So that's basically what their argument is. And I think it's an interesting paper because it leads to a lot of questions about what the function of cell cycle is and what is sort of being, what the outcome of this should be on differentiation, cell differentiation, and ultimately development, and even things like heterochrony. So these simulations are quite interesting. They're doing simulations for single cell division steps. For simplicity, but these are repeated thousands of times to model cell population effects. We compute cell diversity in two ways. First is the number of distinct transcriptomes in the cell population, which is transcriptome diversity, and second is the number of distinct transcriptome clusters, as defined using standard single-cell transcriptomic analysis techniques. That's what we saw in that graph, where they had the transcriptome counts, or the copy number counts, and then they also showed the different things that can be in the transcriptome. And so you just basically model the genome as genes of different length. You model the cell cycle as certain lengths, and then you see which ones are possible within that interval. So it's very, very simple simulation, numeric simulation. And so this is the output here, where the filter is off versus when the filter is on. But actually, when the filter is on, you get more clusters, at least when the cell cycle is around 5 in duration versus later or earlier. And then, so transcriptome diversity actually is affected by being off versus clusters being affected when it's on. And then this shows like this is a t-SNE analysis of the simulation, where you have 2 clusters, 40 clusters, and 25 clusters. In a lineage tree example, this is where we have scenario 1, which is fast, scenario 2, which is slow, fast, and scenario 3, which is slow, fast as well. What they mean by that is, so cells with a longer cell cycle duration, the blue lineage, generates fewer progeny with respect to the cells with a short cell cycle duration of 1 hour, which is the gray lineage. And so the blue, again, is the cell cycle duration, and the gray is the cells of the short cell cycle duration. So these longer cell cycle durations generate fewer progeny. The slower cells contribute more to the diversity observed in the population. So increasing cell cycle duration increases cell diversity, but also limits the number of progeny generated. So we see that in fast, we have 2 sublineages that form the blue and the gray. They're symmetrical. When we have a slow fast scenario, when we slow down the division rate of the blue, we only have 4 cells versus 8 cells at the end of the simulation. So for the fast scenario, transcriptional diversity is really low. It increases with the slow fast scenario, and then the proportion of blue cells and the cell percentage here, the cells that survive this division process, is almost incompletely blue, and the transcriptional diversity is high. Whereas in slow fast, in this case, the gray cells dominate. The transcriptional diversity is also high. In this case, the cell percentage is equal in the fast, but the transcriptional diversity is low. So when these lineages are differential in terms of their strategy, basically they're going to be either outcompeted one way or another, and transcriptional diversity will be high. When they're equivalent in terms of both going fast, they're equivalent in terms of their numbers, their percentage of the population, but the transcriptional diversity is really low. Okay, so that's that paper. The second paper is this core control principles of the eukaryotic cell cycle. So now we go from this idea of transcriptional filtering to this idea of control principles for the cell cycle itself. So that makes sense because we want to know what's underlying this phenomena. And it involves controlling cell cycle. So one of the things at the heart of eukaryotic cell cycle are these cyclin-dependent kinases, or CDKs. Different cyclin CDK complexes initiate DNA replication and mitosis, which is when the DNA is replicated and then when the cell divides. However, the principle on which cyclin CDK complex is organized, the temporal order of cell cycle events, are contentious. So there are two competing models here. And we don't really know how these things are ordered temporally and how they're controlled. The first model is this SCDKs and MCDKs are functionally specialized. So SCDKs are these different cyclin CDK for DNA replication. So one model proposes that SCDKs and MCDKs are functionally specialized. So the SCDKs are these DNA replication CDKs, and the MCDKs are mitosis related. So the ones involved in DNA replication, the ones involved in mitosis, are functionally specialized with substantially different substrate specificities to execute different cell cycle events. A second model proposes that SCDKs and MCDKs are redundant with each other, with both acting as sources of overall CDK activity. In this model, increasing CDK activity rather than CDK substrate specificity orders cell cycle events. So it's actually activity or specificity in the second model. In the first model, it's the idea that you need specificity. Here we reconcile these two views of core cell cycle using phosphoproteomic assays of in vivo CDK activity in fission yeast. We find that SCDK and MCDK substrate specificities are remarkably similar, showing that SCDKs and MCDKs are not completely specialized for S phase and mitosis alone. So these are the DNA replication at S phase and the cell division at mitosis. So we find that there's not a complete specialization of these substrates, that they're interchangeable, but there are also ones that are specialized for each part of cell cycle. And this is important because if you think about how cell cycle might work, you have a bunch of substrates that are occupied in one phase, and then you need to have them occupiable for the next phase if they're interchangeable. If they're not, then they're maybe specialized for those specific phases. And this is something that maybe can control timing. If you have saturation of your substrate, it shortens the length of time you have, or it sort of limits the amount of time you have in cell cycle. So this is kind of an important point and just something to think about. Normally, SCDK cannot drive mitosis, but can do so when protein phosphatase 1 is removed from the centrosome. Thus, increasing SCDK activity in vivo is sufficient to overcome substrate specificity differences between SCDK and MCDK, and allows SCDK to carry out MCDK function. So you have this interchangeability. And there is a lot of, increasing activity can overcome substrate specificity in certain conditions. And so we have this sort of, I guess you could say, biological adaptability or even biological robustness. Therefore, we unite the two opposing views of cell cycle control, showing that the core cell cycle engine is largely based on quantitative increases in CDK activity through the cell cycle. So we have these increases in CDK activity, and that's what drives different parts of the cell cycle and its timing, combined with minor insurmountable qualitative differences in catalytic specialization. So that means that there is also a role for specialization, and that also drives timing, the sort of the unfolding of that process, and so forth. So this kind of goes through some of their data. It's a pretty deep paper in terms of technical specificity. So for this group, I don't think that you're going to get a lot out of it by going through the details. I think that this does kind of complement this other paper, and then it gives a mechanism for what's going on there. The other paper is just kind of like looking at the mRNA and saying, what is the expression level? We can hypothesize that shorter timing will lead to shorter amounts of transcription, and we can confirm that with a data set. This shows you the actual mechanisms behind it and some of the biochemistry. So this is an interesting set of papers, and I think it has a lot of consequences for thinking about cell division and some of the variation we see across species and across developmental time. So thank you for paying attention, and I hope you learned something. Well, so yeah, I guess that's pretty much it for today. Do we have any other comments or questions? I can send out the papers, and yeah. Otherwise, I think I don't know where Himanshu is today, but we'll continue next week with the community period. And I want to remind people to have your that Sushmanth and Himanshu I want them to give presentations. I hope I mentioned that last week I mentioned it in the email when we started, but that we have I want basically to go over your proposals and mark down like kind of what where you are with the proposal, say what you're wanting to do with the summer, and just summarizing it and putting in slides. And then at the end of the summer, we will go over those and replace the slides where you didn't achieve what you wanted to, or you achieved much more than you wanted to, or that you actually did some work on it, and then you'll present that again. And the point is to show the progress and sometimes the detours that you have to make. So I don't know when we'll present those. Probably not, maybe not next week, but the week after. And that'll be in the coding period, but it'll be fine. And then it looks like Sushmanth is, just continuing to go on with his coding work. So thank you, Sushmanth. That looks great. And I know the audio is a little muddled, but it's fine. I think we got the idea of what you're doing. So,
Bradley,
yeah.
I'll give you one more historical example.
Okay.
You remember the hoopla over cloning of animals?
Oh yeah. Yeah.
Yeah. Well, it was both a success and a disaster because the animals were cloned nicely, and then they died of old age at a very early age.
Yeah.
Okay. And I haven't seen any advertisements for cloning your pets lately.
Oh yeah. Well, it's yeah.
Okay, so these things, cloning looked very good on paper, and it seemed to work. Then it screwed up.
Oh yeah.
Okay, so all these manipulations, gene manipulation, cell manipulation, etc. We have to tread carefully.
Yeah. All right. Well, thanks for attending. Talk to everyone next week. Bye.
Thank you.
Yep.
Bye.
Bye.